Hi, good evening. Welcome back to my lecture classes. I'm Dr. Bobby. This is New Age Cosmetic-Wide EdTech Healthcare Consultancy, and I welcome all of you to the daily lectures in biology and chemistry. Today we're dealing with protein folding and processing, which is part of the fundamental processes. We were discussing RNA synthesis and processing in the previous lecture. Today we are on to protein synthesis and processing. We wish to extend our courtesies to National Library of Medicine, the National Center for Biotechnology and Information, for the invaluable inputs and their literature for us to study. I welcome all students to attend this lecture. May take screenshots and make necessary notes, which could be useful for your examinations. So going ahead with the subject, protein folding and processing. Translation completes the flow of genetic information within the cell. The sequence of nucleotides and DNA has now been converted to the sequence of amino acids in a polypeptide chain. The synthesis of a polypeptide chain, however, is not equivalent to the production of a functional protein. So to be useful, polypeptides must fold into distinct three-dimensional conformation, and in many cases, multiple polypeptide chains must assemble into a functional complex. In addition, many proteins undergo further modifications, including cleavage and the covalent attachment of carbohydrates and lipids that are critical for the function and correct localization of proteins within the cell. Chaperones and protein folding. The three-dimensional conformations of protein result from interactions between the side chains of the constituent amino acids, as reviewed in Chapter 2. The classic principle of protein folding is that all the information required for a protein to adopt the correct three-dimensional conformation is provided by its amino acid sequence. This was initially established by Christian and Vincen's experiments demonstrating that denatured RNAs can spontaneously refold in vitro to its active conformation, as shown in figure 217. Let's have a look at 217. And this is the figure, right, that you might see, right? Please take a screenshot of these as you fold into the matter proper. And the protein folding thus appeared to be a self-assembly process that did not require additional cellular factors. More recent studies, however, have shown that this is not an adequate description of protein folding within the cell. The proper folding of proteins within cells is mediated by the activities of other proteins. Proteins that facilitate the folding of other proteins are called molecular chaperones. Well, as I mentioned, chaperones in one of my previous lectures. As I go through these notes, these are research notes, so each note will be explained as I come to it in each sentence. So here we just mentioned the term chaperones. Well, a chaperone is a helix, is an alpha helix. It has a coiled secondary structure of a polypeptide chain formed by hydrogen bonding between amino acids separated by 4 residues. And the related terminologies would be explained accordingly. And there are ABC transformers. A large family of membrane transport proteins is characterized by a highly conserved ATP binding domain. These are important points and terminology that you need to know. So in between the lectures, I'd be explaining these terms in between, so please take note. Next word would be actin, an abundant 43 kD protein that polymerizes to form cytoskeletal filaments. Actin bundle would be actin filaments that are cross-linked into closely packed arrays. Actin network would be actin filaments that are cross-linked into loose three-dimensional meshworks. Activation energy would be the energy required to raise a molecule to its transition state to undergo a chemical reaction. Active site would be the region of an enzyme that binds substrates and catalyzes an enzymatic reaction. Active transport, the transport of molecules in an energetically unfavorable direction across a membrane coupled to the hydrolysis of ATP or other source of energy. Adaptin would be a protein that binds to membrane receptors and mediates the formation of clathrin-coated vesicles. Adenine, a purine that base pairs with either thymine or uracil. Adenoma, a benign tumor arising from glandular epithelium. Adenylyl cyclase, as an enzyme that catalyzes the formation of cyclic AMP from ATP. Adherence junction, a region of cell-cell adhesion at which the actin cytoskeleton is anchored to the plasma membrane. AKT is a protein serine 309 kinase that is activated by PIP3 and plays a key role in signaling cell survival. Allele is one copy of a gene. Allosteric regulation, the regulation of enzymes by small molecules that bind to a site distinct from the active site, changing the conformation and catalytic activity of the enzyme. Alternative splicing, the generation of different mRNAs by varying the patterns of pre-mRNA splicing. Amino acid, monomeric building blocks of proteins consisting of a carbon atom bound to a carboxyl group, an amino group, a hydrogen atom, and a distinctive side chain. Amino-acyl tRNA synthetase, an enzyme that joins a specific amino acid to tRNA molecule carrying the correct anticodon sequence. Amphipathic would be a molecule that has both hydrophobic and hydrophilic regions. Anaphase would be the phase of mitosis, during which sister chromatids separate and move to opposite poles of the spindle. Anaphase A is the movement of daughter chromosomals towards the spindle poles during mitosis. Anaphase B would be the separation of the spindle poles during mitosis. Anaphase promoting complex, a ubiquitin ligase that triggers progression from metaphase to anaphase by signaling the degradation of cyclin B and cohescence. So we stop the terms at that point now and return to the literature proper, and we'll return as a little more portions are over as our regular period of time. I'll keep elaborating these terminologies so you get to know the subject proper much better, right? Okay. Back to the subject proper. We were at this term of chaperones. The term chaperone was first used by Ron Lasky and his colleagues to describe a protein nucleoplasmin that is required for the assembly of nucleosomes from histones and DNA. Nucleoplasmin binds to histones and mediates the assembly into nucleosomes, but nucleoplasmin itself is not incorporated into the final nucleosome structure. Chaperones thus act as catalysts that facilitate assembly without being part of the assembled complex. Subsequent studies have extended the concept to include proteins that mediate a variety of other assembly processes, particularly protein folding. It is important to know that chaperones do not convey additional information. I repeat, it is important to know that chaperones do not convey additional information required for folding of polypeptides into their correct three-dimensional conformations. The folded conformation of a protein is determined solely by its amino acid sequence. Rather, chaperones catalyze protein folding by assisting the self-assembly process. They appear to function by binding to and stabilizing unfolded or partially folded polypeptides that intermediates along the pathway leading to the final correctly folded state. In the absence of chaperones, unfolded or partially folded polypeptide chains would be unstable within the cell, frequently folding incorrectly or aggregating into soluble complexes. The binding of chaperones stabilizes these unfolded polypeptides, thereby preventing incorrect folding or aggregation and allowing the polypeptide chain to fold into its correct conformation. A good example is provided by chaperones that bind to nascent polypeptide chains that are still being translated on ribosomes, thereby preventing incorrect folding or aggregation of the amino terminal portion of the polypeptide before synthesis of the chain is finished. We go into figure 717 for purpose of continuity, right? So this is figure 717, which is seen here, right? That's it, and we come back to subject matter proper, right? And presumably this interaction is particularly important for proteins in which the carboxy terminus, the last to be synthesized, is required for correct folding of the amino terminus. In such cases, chaperone binding stabilizes the amino terminal portion in an unfolded conformation until the rest of the polypeptide chain is synthesized and the completed protein can fold correctly. Chaperones also stabilize unfolded polypeptide chains during the transport into subcellular organelles, for example, during the transfer of proteins into mitochondria from the cytosol. That will be figure 718. That's the other one. So action of chaperones during protein transport. A partially unfolded polypeptide is transported from the cytosol to a mitochondrion. Cytosolic chaperones stabilize the unfolded configuration. Mitochondrial chaperones facilitate transport and subsequent further on to details of that would be, this is the chaperone, right? And so we're going back. And okay, that's it about that. And then we continue with the subject matter proper. Proteins are transported across the mitochondrial membrane in partially unfolded conformations that are stabilized by chaperones in the cytosol. Chaperones within the mitochondrion then facilitate transfer of the polypeptide chain across the membrane and its subsequent folding within the organelle. In addition, chaperones are involved in the assembly of proteins that consist of multiple polypeptide chains, the assembly of macromolecular structures, example nucleoplasmin, and as discussed later in this chapter, in the regulation of protein degradation. So figure 717, as we already saw, action of chaperones during translation. Let's look at this. Action of chaperones during translation. Chaperones bind to the amino N-terminus of the growing polypeptide chain, stabilizing it in an unfolded configuration until synthesis of the polypeptide is complete. The completed protein is then released from the ribosomes and is able to fold into its correct three-dimensional conformation, right? And as we return to the original chapter, we have info that states the action of chaperones during translation, that it binds to the amino N-terminus of the growing polypeptide chain, stabilizing it in an unfolded configuration until synthesis of the polypeptide is completed. The completed protein is then released from the ribosome and is able to fold into its correct three-dimensional conformation. Now, this figure that was already shown to you. It states that the action of chaperones during protein transport, a partially unfolded polypeptide is transported from the cytosol to a mitochondrion. Cytosolic chaperones stabilize the unfolded folded configuration. Mitochondrial chaperones facilitate transport, and subsequently more would be the additional info on that. And subsequent folding of the polypeptide chain within the organelle from protein folding and processing a cell. This is from there. We're going back now. And now many of the proteins now known to function as molecular chaperones in table 7.2 were initially identified as heat shock proteins. So let's see what are heat shock proteins, right? Now if we go back to the glossary, we see heat shock proteins, a highly conserved group of chaperone proteins expressed in cells exposed to elevated temperatures or other forms of environmental stress. So from now on, as I read from these top subjects, there'll be terms that you don't know. So the moment we come to a term like that, I'll switch over to the glossary and give you an explanation on those terms, then go back to the subject proper and we continue from there, right? So that gives a continuity, you get an understanding, you get a basis of what you're studying. Because unless you know the basics, you're not going to understand what's happening, right? So that's about heat shock proteins. Let me repeat that. A highly conserved group of chaperone proteins expressed in cells exposed to elevated temperatures or other forms of environmental stress. Other terminologies would be helicase, an enzyme that catalyzes the unwinding of DNA. Hemicellulose, a polysaccharide that cross-links cellulose microfibrils in plant cell walls. Hemidesmosome, part of the region of contact between cells and the extracellular matrix at which keratin filaments are attached to integrin, heterochromatin, condensed transcriptionally inactive chromatin. Okay, going back. To the subject matter proper, right? We were at chaperones, right? And we see the figure there, we see the figure here, right? Here we were with heat shock proteins. I hope you've taken down your notes, right? Well, it's a group of proteins expressed in cells that have been subjected to elevated temperatures or other forms of environmental stress. So remember the word HSP. The HSP, so heat shock proteins abbreviated as HSP, which are highly conserved in both prokaryotic and eukaryotic cells, are thought to stabilize and facilitate the free falling of proteins that have been partially denatured as a result of exposure to elevated temperature. However, many members of the heat shock protein family are expressed and have essential cellular functions under normal growth conditions. These proteins serve as molecular chaperones, which are needed for polypeptide folding and transport under normal conditions, as well as in cells subject to environmental stress. Molecular chaperones. Let's check the table here. Well, HSP70 is DNA case in prokaryotes. Eukaryotes, it's HSC73 cytosol, BIP endoplasmic reticulum, SSC1 mitochondria, CDHSP70 chloroplast. HSP60 will be GRO-EL and Tri-C cytosol, HSP16 mitochondria, CPN16 chloroplast, and HSP90 would be the HTPs in prokaryotes, and in eukaryotes it will be HSP90 cytosol and GRP94 endoplasmic reticulum. Right? These are the molecular chaperones. The HSP70 and HSP60 families of heat shock proteins appear to be particularly important in the general pathways of protein folding in both prokaryotic and eukaryotic cells. The proteins of both families function by binding to unfolded regions of polypeptide chains. They are members of the HSP family, stabilize unfolded polypeptide chains during right, and moving on to the next paragraph. Members of the HSP60 family, also called chaperones, facilitate the folding of proteins into their native conformations. Each chaperone consists of 14 subunits of approximately 60 kilodaltons kd each, arranged in two stacked rings to form a double donut structure, which is shown in figure 719. Let's have a look at that. Right, permission to produce this figure in this web version of the cell, a molecular approach addition is either pending or has not been granted. So we have that figure is not there. We come to that later, not a problem. So unfolded polypeptide chains are from the cytosol, are shielded from the cytosol by being bound within central gravity of the chaperone in cylinder. In this isolated environment, protein folding can proceed while aggregation of unfolded segments of the polypeptide chain is prevented by their binding to the chaperonin. The binding of unfolded polypeptides to the chaperonin is a reversible reaction that is coupled into the hydrolysis of ATP as a source of energy. ATP hydrolysis thus drives multiple rounds of release and rebinding of unfolded regions of the polypeptide chains to the chaperonin, allowing the polypeptide to fold gradually into correct conformation. Right? So that is the structure of 719. The figure is yet to be available. We'll check on that later, but then I'll give you the details on the theory. The structure of a chaperonin, GRO-EL, a member of the HSP60 family, is a porous cylinder composed of two stacked rings. Each ring consists of 7 subunits, right? So that will be details on that. Right, and in some cases, members of the HSP70 and HSP60 families have been found to act together in a sequential fashion. For example, HSP70 and HSP60 family members act sequentially during the transport and of proteins into mitochondria and during the folding of newly synthesized proteins in E. coli. And let's check E. coli on this figure. And yes, we have the picture of E. coli, right? And if you see here, this is how it is. That's it about that figure there. And first an HSP70. Chaperone stabilizes nascent polypeptide chains until protein synthesis is completed. The unfolded polypeptide chain is then transferred to an HSP60 chaperone, within which protein folding takes place, yielding a protein correctly folded into its functional three-dimensional conformation. Well, members of the HSP 70 and HSP60 families are found in the cytosol and subcellular organelles, for example in mitochondria of eukaryotic cells as well as in bacteria. Table 7.2. We'll see the table later. We'll have a look at it. This is the table of right, table 7.2. Chaperone proteins, protein family prokaryotes, eukaryotes. I just about read this to you, right? And we go back. So the sequential action of HSP70 and HSP60 appear to represent a general pathway of protein folding. An alternative pathway for the folding of some proteins in the cytosol and endoplasmic reticulum may involve the sequential actions of HSP70 and HSP90 family members, although the functions of HSP90 is not yet well understood. This is figure 77.2, which is a depiction of sequential action of HSP70 and the HSP60 chaperones. Right, and the sequential action of HSP70 and HSP60 chaperones of the HSP70 family bind to and stabilize unfolded polypeptide chains during translation. The unfolded polypeptide is then transferred to chaperones with the HSP60 family, within which protein folding takes place. ADP hydrolysis is required for release of the unfolded polypeptide from HSP70 as well as for folding within HSP60. And we go back to the subject proper, right? Enzymes and protein folding. In addition to chaperones, which facilitate protein folding by binding to and stabilizing partially folded intermediates, cells contain at least two types of enzymes that catalyze protein folding by breaking and reforming covalent bonds. The formation of disulfide bonds between cysteine residues is important in stabilizing the folded structure of many proteins, as we see in figure 216. Disulfide bond between cysteine residues. Just remember the diagram, take a screenshot, you should be okay. For proteins that contain multiple cysteine residues, protein disulfide isomerase, PDI, plays an important role by promoting rapid exchanges between paired disulfides by allowing the protein to attain the pattern of disulfide bonds that is compatible with its stably folded conformation. Disulfide bonds are generally restricted to secreted proteins and some membrane proteins because the cytosol contains reducing agents that maintain cysteine residues in their reduced SH form, thereby preventing the formation of disulfide SS linkages. In eukaryotic cells, disulfide bonds form in the endoplasmic reticulum, in which an oxidizing environment is maintained. Consistent with the role of disulfide bonds in stabilizing secreted proteins, the activity of PDI in the endoplasmic reticulum is correlated with the level of protein secreted in different types of cells. And this is figure 7.21, which shows the action of protein disulfide isomerase, which catalyzes the breakage and rejoining of disulfide bonds, resulting in exchanges between paired disulfides in a polypeptide chain. The enzyme forms a disulfide bond with the cysteine residue of the polypeptide and then exchanges its paired disulfide with another cysteine residue In this example, PDI catalyzes the conversion of two incorrect disulfide bonds, 1, 2, and 3, 4 to the correct, pairing 1, 3, and 2, 4. Alright, and the second enzyme that plays a role in protein folding catalyzes the isomerization of polypeptide bonds that involve proline residues. That's in 7.22. That's the table. Excuse me. That's a figure. Let's see that. That's the figure there. The action of peptidyl-prolyl isomerase. Peptidyl-prolyl isomerase catalyzes the isomerization of peptide bonds. Right, continuing from where we last stopped, is a good example is provided by chaperones that bind to a nascent polypeptide, chains that are still being translated on ribosomes, thereby preventing incorrect folding or aggregation of the minor terminal portion of the polypeptide before synthesis of the chain is finished. Presumably, this interaction is particularly important for proteins in which the carboxy terminus, the last to be synthesized, is required for correct folding of the amino terminus. In such cases, chaperone binding stabilizes the amino terminal portion in an unfolded conformation until the rest of the polypeptide chain is synthesized and the complete protein can fold correctly. Chaperones also stabilize unfolded polypeptide chains during the transport into subcellular organelles, for example, during the transfer of proteins in the mitochondria from the cytosol. Proteins are transported across the mitochondrial membrane in partially unfolded conformations that are stabilized by chaperones in the cytosol. Chaperones within the mitochondrion then facilitate transfer of the polypeptide chain across the membrane and its subsequent folding within the organelle. In addition, chaperones are involved in the assembly of proteins that consist of multiple polypeptide chains, the assembly of macromolecular structures, nucleoplasmin, and as discussed earlier, in the regulation of protein degradation. Alright, we finished all this. Now we go down to the latter portion. Right? We could go even further because we just finished all this. Now we'll go on to enzymes and protein folding. In addition to chaperones, which facilitate protein folding by binding to and stabilizing partially folded intermediates, cells contain at least two types of enzymes that catalyze protein folding by breaking and reforming covalent bonds. The formation of disulfide bonds between cysteine residues is important in stabilizing the folded structure of many proteins. Now, protein disulfide isomerase, which was discovered by Christian and Fincher in 1963, catalyzes the breakage and reformation of these bonds. For proteins that contain multiple cysteine residues, protein disulfide isomerase (PDI) plays an important role by promoting rapid exchanges between paired disulfides, thereby allowing the protein to attain the pattern of disulfide bonds that is compatible with its stably folded conformation. Disulfide bonds are generally restricted to secreted proteins and some membrane proteins because the cytosol contains reducing agents that maintain cysteine residues in their reduced SH form, thereby preventing the formation of disulfide SS linkages. In eukaryotic cells, disulfide bonds form in the endoplasmic reticulum in which an oxidizing environment is maintained. Consistent with the role of disulfide bonds in stabilizing secreted proteins, the activity of PDI in the endoplasmic reticulum is correlated with the level of protein secretion in different types of cells. The action of protein disulfide isomerase, PDI, catalyzes the breakage and rejoining disulfide bonds, resulting in exchanges between paired disulfides in a polypeptide chain. The enzyme forms a disulfide bond with a cysteine. The second enzyme that plays a role in protein folding catalyzes the isomerization of peptide bonds that involve chloride proline residues. Proline is an unusual amino acid in that the equilibrium between the cis and trans conformation of peptide bonds that precede proline residues is only slightly in favor of the trans form. In contrast, peptide bonds between other amino acids are almost always in the trans form. Isomerization between the cis and trans configurations of prolyl peptide bonds, which could otherwise represent a rate-limiting step in protein folding. Is catalyzed by the enzyme peptidyl prolyl isomerase. This enzyme is widely distributed in both prokaryotic and eukaryotic cells and can catalyze the folding of at least some proteins. However, its physiologically important substrates and role within cells are yet to be determined. This is figure 712, which we've already seen. Just have a look. Action of peptidyl prolyl isomerase catalyzes the isomerase peptide bond that involves proline between the cis and trans conformations. The action of peptidyl prolyl isomerase, prolyl isomerase catalyzes the isomerase peptide bonds that involve proline between the cis and trans conformation. Protein cleavage. Cleavage of the polypeptide chain polyproteolysis is an important step in the maturation of many proteins. A simple example is removal of the initiated methionine from the amino terminus of many polypeptides, which occurs soon after the amino terminus of the growing polypeptide chain emerges from the ribosome. Additional chemical groups such as diacetyl groups or fatty acid chains discussed shortly are then added to the amino terminal residues. Proteolytic modifications of the amino terminus also play a part in the translocation of many proteins across membranes, including secreted proteins in both bacteria and eukaryotes, as well as proteins destined for incorporation into the plasma membrane. Lysosomes, mitochondria, and chloroplast amino terminal sequence that are removed by proteolytic cleavage as the protein crosses the membrane. For example, amino terminal signal sequence is usually about 20 amino acids long. Targeted secreted proteins to the plasma membrane bacteria or to the endoplasmic reticulum or eukaryotic cells, while translation is still in progress. The signal sequence, which consists predominantly of hydrophobic amino acids, is inserted into the membrane as it emerges from the ribosomes. The remainder of the polypeptide chain passes through a chain in the membrane as translation proceeds. The signal sequence is then cleaved by a specific membrane protease, signal peptidase, and the mature protein is released. In eukaryotic cells, the translocation of growing polypeptide chains into the endoplasmic reticulum is the first step in targeting proteins for secretion. Incorporation into the plasma membrane and incorporation into lysosomes. The mechanisms that direct the transport of proteins to these destinations, as well as the role of other targeting sequences in directing the import of proteins into mitochondria and chloroplasts, will be discussed in the following chapters of 9 and 10 as we proceed. Now, the role of signal sequences in membrane translocation. So let's see this figure. That is about signal sequence. Right? See the role of translocation of polypeptide chains across the plasma membrane of bacteria or into the endoplasmic reticulum of eukaryotic cells shown here. The signal sequence is a stretch of hydrophobic amino acids at the amino terminus of the polypeptide chain inserted into a membrane channel as it emerges from the ribosome. The rest of the polypeptide is then translocated through the channel, and the signal sequence is cleaved by the action of signal peptidase, releasing the matured translocated protein. And in other important instances of proteolytic processing, active enzymes or hormones are formed via cleavage of larger precursors. Insulin, which is synthesized as a longer precursor polypeptide, is a good example. Insulin forms by two cleavages. The initial precursor proinsulin contains an amino terminal sequence that targets the polypeptide chain to the endoplasmic reticulum. Removal of the signal sequence during transfer to the endoplasmic reticulum yields a second precursor called proinsulin. This precursor is then converted to insulin, which consists of two chains held together by disulfide bonds by proteolytic removal of an internal peptide. Other proteins activated by similar cleavage processes include digestive enzymes and the proteins involved in blood clotting. Figure 724. This is about proteolytic processing of insulin. The mature insulin molecule consists of two polypeptide chains A and B, proinsulin and insulin, joined by disulfide bonds. It is synthesized as a precursor polypeptide, proinsulin, containing an amino terminal signal sequence, and that is cleaved during transfer of the growing polypeptide chain to the endoplasmic reticulum. This cleavage yields a second precursor proinsulin, which is converted to insulin by further proteolysis, removing the internal connecting polypeptides. That's it for this lecture.